Welcome to the Deep Dive. Today is Thursday, September 11, 2025. Each week, we scour the latest research to bring you what we think are the most impactful discoveries in longevity science.
Right.
Now, casting our net this week, I gotta be honest with you, it feels like a fairly typical week out there in the field.
hmm
We've got two new, pretty intriguing papers that definitely caught our eye. But, it's mostly solid, incremental research.
Yeah.
Not quite those huge paradigm-shifting, earth-shattering breakthroughs we're always kind of hunting for.
And that's really the nature of scientific progress, isn't it? I mean, in a field moving as fast as longevity, it's easy to get swept up in the latest headlines.
Totally.
But true understanding often comes from really appreciating the foundations, the monumental work that laid the groundwork for everything we're seeing today.
Yeah,
Without that context, it's just much harder to figure out what's truly significant now.
I completely agree. And that's exactly why, for today's discussion, we're taking a slightly different approach. Because it is a more typical week of these sort of incremental findings, we thought let's bring back a true classic, a foundational paper by the incredible Cynthia Kenyon from back in 2011.
Ah, yes. Excellent choice.
This work, published in Royal Society Publishing, it doesn't just describe a discovery, it beautifully captures the moment, a specific longevity gene pathway was first found, a discovery that fundamentally changed how we even think about aging.
It really did.
And we'll be using the Norn Study Group papers and their analysis to kind of guide our discussion of this landmark work.
That's a great plan. Revisiting Kenyon's paper, it gives us such crucial context for evaluating today's, incremental progress. It reminds us of the sheer scale of impact a single well-executed line of inquiry can have, how it can just pivot an entire field.
Absolutely. But don't worry, we're not just looking backward today. We'll also dive into two recent papers that, despite being maybe more incremental in isolation, still offer some really fascinating insights and potential future directions.
Okay, what do we got?
So first up, we have the aging factor, EPS8, induces disease-related protein aggregation through RAC signaling hyperactivation. That's from the University Hospital Cologne, published just last week, September 4th.
Okay, EPS8 and aggregation. Got it.
And then we'll explore platelet factor 4, PF4, regulates hematopoietic stem cell aging, which just came out this Monday, September 8th, from the University of Illinois at Chicago.
PF4 and stem cells.
Right.
Both sound like solid pieces of research, like you said, adding important little pieces to this ever-growing understanding of aging.
Exactly. So our mission today is pretty clear. We're here to extract the most important nuggets of knowledge, the insights from all these sources, the historical, the cutting edge. We want to help you quickly understand what's most important, maybe where the real breakthroughs might be hiding, and really crucially, where to apply a healthy dose of scientific skepticism.
Always important.
Think of this as your shortcut to being well-informed, complete with maybe some surprising facts and that critical analysis, so you can walk away with a deeper appreciation for this incredible field. Okay, let's unpack this and dive in.
Let's do it.
Okay, let's start with that classic Cynthia Kenyon's 2011 paper, the first long-lived mutant's discovery of the insulin IGF-1 pathway for aging. What's so fascinating is that even now, in 2025, this paper is still incredibly relevant. I mean, it's almost a quarter century after the initial discoveries it talks about.
Yeah.
What was the scientific landscape actually like before Kenyon's work? Why does her contribution continue to resonate so profoundly?
Well, before Kenyon's work really hit, the prevailing view of aging was honestly that it was just this intractable passive process.
Right, things just break down.
Exactly. The idea was simply that organisms just wear out over time. Like an old machine. It was seen as something inevitable, kind of like entropic decay, definitely not something genetically regulated or, modifiable. There was even a really strong school of thought among evolutionary biologists arguing against the whole idea of regulated aging.
Really? Why?
Well, they suggested that natural selection couldn't really act on processes that happen after reproduction. So, in their view, aging was biologically irrelevant and therefore unregulatable. This created a powerful bias against even looking for genetic controls of aging. It was pretty much seen as a research dead end.
Wow. So it was almost like saying, things just fall. There's no deeper force like gravity. And then along comes Cynthia Kenyon. Her background wasn't even in aging research itself, right? She came from developmental biology. How did that shape how she looked at aging?
That background was absolutely pivotal. Yeah, Kenyon was a developmental biologist at UCSF, and in that field, researchers were just used to seeing these universal, evolutionarily conserved regulatory mechanisms.
Like Hox genes.
Exactly. Think about the famous Hox genes, the
Mhm.
precisely patterned body development across vastly different species, you know, from flies all the way to humans. This experience led her to this, well, profound hypothesis. If development, this incredibly complex biological process, was so elegantly regulated by genes, why not aging?
Huh. Makes sense when you put it like that.
She envisioned these universal aging mechanisms, controlled by regulatory genes, whose activities could potentially be, like, dialed up or down to alter lifespan.
That's a truly revolutionary shift in perspective. So to her, aging wasn't some biological dead end, but she actually called it a fantastic opportunity to explore the unknown and perhaps discover something new and important.
Yeah.
That kind of scientific curiosity, that willingness to challenge these really deeply ingrained assumptions, must have been incredibly vital. Like she was almost a lone voice against this chorus of naysayers.
It really was. It was a very challenging path initially. I mean, many molecular biologists at the time really did consider aging research a backwater. Oh, yeah. Colleagues openly warned her that she would fall off the edge of the earth if she pursued it.
Wow.
But her fundamental belief, informed by her work on other biological processes, was that phenomena that seemed haphazard often turned out to be precisely controlled by genes. This mindset, it allowed her to see aging not as this futile problem, but as fertile ground for groundbreaking discovery. It really required immense courage and conviction.
So where did she even begin this exploration? The sources point to the small roundworm, Caenorhabditis elegans, or C. elegans. This little nematode, it's been a superstar in biology, but how did it first get into the longevity story even before Kenyon got directly involved?
Right, the story really starts even earlier with some crucial foundational steps taken by researchers like Michael Klass, way back in 1983. Klass was a postdoc in David Hirsch's lab at the University of Colorado, and he was among the very first to systematically screen for long-lived mutants in C. elegans, which in itself was quite a radical departure from that prevailing skepticism about regulated aging.
And he found some interesting stuff even then, didn't he? Like glimmers of environmental effects on longevity.
Yes, exactly. Klass observed that C. elegans lived longer and, interestingly, had fewer progeny when subjected to dietary restriction. Now, that phenomenon itself was first seen in rodents way back in the 1930s.
Right.
He also showed that worms, being ectotherms, lived longer at lower temperatures. But what's even more intriguing, I think, is that he found worms carried a kind of memory of their childhood temperature that affected their adult lifespan.
Memory.
It's a molecular mechanism that honestly still isn't fully explained even today. But these are really early hints that lifespan wasn't just some fixed duration.
That's wild, a temperature memory affecting adult life. But his actual screen for long-lived genes, the very first one, it didn't really seem to yield what he was looking for, did it? It almost reinforced the skepticism.
That's a critical point, yeah. Klass used this clever strategy involving temperature-sensitive FIR15 mutants. These prevented reproduction at high temperatures, which let him screen for longevity without the animals just overpopulating the plates.
Right.
He identified 8 long-lived families. However, after more observation, he concluded that most of these mutants were long-lived simply because they were eating less, essentially, a form of dietary restriction.
Ah, okay.
His famous, albeit initially, well, discouraging conclusion was that specific lifespan genes are extremely rare, or alternatively, lifespan is controlled in a polygenic fashion.
Yeah.
This just perfectly illustrates that initial skepticism and the difficulty in distinguishing true longevity genes from these indirect effects like caloric restriction. It was a major conceptual roadblock back then.
So the field was still pretty much entrenched in that wearing out idea, even when faced with potential evidence against it. But Klass's pioneering work wasn't totally abandoned. Tom Johnson picks up the thread, right, and eventually finds the first true longevity gene.
That's where Tom Johnson from the University of Colorado comes in during the late 80s. He initially approached the problem a bit differently. He crossed 2 different C. elegans strains, Bristol and Bergerac, and created these recombinant inbred lines.
Okay.
And he found that some of these lines lived much longer than others, which strongly suggested that genetic polymorphisms, just natural variations in DNA, could influence lifespan. This was a critical step in establishing a genetic basis for lifespan variation.
And then, importantly, he circled back to Klass's work and found a strain that really seemed to defy that simple caloric restriction explanation.
Yes, exactly. He subsequently outcrossed some of Klass's original mutants and recovered a specific strain, H1 H546. And this strain ate normally, but lived a remarkable 40–55% longer.
Wow.
This was incredibly significant because it seemed to finally decouple longevity from dietary restriction, suggesting a direct genetic influence.
Okay, but here's where it takes a really fascinating turn, though. This age 1 mutant had sharply reduced fertility, and that immediately brought back the sort of trivial explanation of the evolutionary trade-off theory. The idea that, reduced reproduction just shunts resources to somatic maintenance, making the animal live longer. Johnson himself even considered this, right? That age 1's main effect might be on fertility, not aging directly. How did they sort that out?
That was indeed a major conundrum. It really challenged the interpretation of the age 1 discovery. However, crucially, in 1993, Johnson performed another key experiment. He successfully crossed away the fertility defect of age 1,
Ah,
and the worms still live long.
okay, that's definitive.
It was a definitive piece of evidence, finally establishing age 1 as a true longevity gene, with its effects clearly distinct from that simple trade-off with reproduction. It was a huge conceptual leap forward for the field.
So that's a massive step. It took the idea of regulated aging from just a theoretical concept to something tangible, something genetic. And this is when Cynthia Kenyon really enters the scene directly, taking on this reproductive trade-off theory head-on.
Exactly. Kenyon had heard Johnson speak at conferences and found the age 1 discovery extremely exciting. But she too was skeptical of the simple idea that just reducing reproduction automatically led to longevity. She felt it was, well, too simplistic and wanted to understand if there was a deeper regulated mechanism really at play.
So she decided to test the reproductive trade-off theory directly herself. This led to her student Ramon Tab-Tang's laser ablation experiment in 1992. Can you walk us through that? It sounds like a really elegant experiment. And what was the conclusion?
Certainly, yeah, it was quite elegant. To directly challenge that trade-off theory, Ramon Tab-Tang used a finely focused laser to basically ablate or destroy the reproductive precursor cells of normal worms.
Okay.
This made them completely sterile. The hypothesis was pretty straightforward. If the trade-off theory was correct, these sterile worms, now completely free of the huge energetic demands of making eggs and sperm, should live longer.
Makes sense. So what happened?
Well, the result was a clear refutation of that simple trade-off.
Huh.
Precisely. The result was definitive. The sterile worms had a completely normal lifespan.
No difference at all.
None. This was a definitive debunking of the simple reproductive trade-off. It showed that just freeing up resources from germ cells wasn't automatically getting redirected into longevity pathways. It reinforced some earlier findings with those FUR15 mutants, but this was just a much more direct and impactful demonstration. It unequivocally told them that something else, something beyond simple resource allocation, had to be going on.
Wow. Okay, so with the simple reproductive trade-off theory kind of put to bed, Kenyon felt much more confident pursuing her own screen for long-lived mutants, even with all the resistance from students and colleagues who still thought aging was this backwater.
Right.
This is where the DAF2 discovery really opened the floodgates, isn't it?
This is where their strategy became really clever, drawing inspiration from existing C. elegans biology. They decided to leverage the Dauer state.
Dauer, right, the German word for enduring.
Exactly. It's this fascinating diapause state that C. elegans can enter under harsh conditions, like low food or crowding. It's basically a quiescent, extremely long-lived, non-feeding juvenile stage. And mutations in genes like DAF2 were already known to make worms Dauer constitutive.
Meaning they just enter that state automatically.
Right, they prematurely enter this long-lived state, even with plenty of food around.
So their idea was, let's mutagenize worms with a temperature-sensitive DAF2 mutation, and then just look for adult worms that live surprisingly long, kind of using the Dauer pathway as a proxy for finding longer life.
It was a brilliant approach. And what happened next was just one of those strokes of scientific serendipity. Ramon Tab-Tang, who was actually a rotation student at the time.
The laser guy.
The same one.
Yeah.
He observed that the controlled DAF2 anomers, the ones that were supposed to be the baseline for measuring when other mutants died, they just weren't dying.
Huh.
He kept asking, apparently, what's going on? These worms are still alive.
They're not dying. That must have been an incredible moment in the lab. Just disbelief and excitement.
It absolutely was. They had pretty much accidentally discovered their first truly long-lived mutant. When they cultured these DAF2 mutants at 20 degrees C, they lived more than twice as long as normal wild-type worms.
Twice as long.
Yeah. And critically, they weren't just like barely existing, they were active and healthy. Kenyon described it as magical, but also a little creepy because, they really should have been dead.
I can imagine.
This thrilling discovery made it much easier, as you can imagine, to attract students to work on aging. It really turned it from that backwater into a vibrant, exciting field almost overnight.
Oh, okay, so here's where the detective work gets really interesting. Unraveling the genetic pathway itself, they now had DAF2 as a key player. What came next in understanding its mechanism, and how did they separate its role in development in dower from its role in adult aging?
Right. The first critical insight was realizing that the severe reduction in DAF2 activity that triggered dower formation during development was actually distinct from the milder reductions that simply extended adult lifespan.
Uh,
They showed that worms cultured continuously at 20 degrees C, where they grew normally to adulthood, still lived incredibly long. And later RNAi analysis confirmed that the wild-type DAF2 gene acts exclusively during adulthood to affect aging, separately from its developmental role in dower formation.
so it wasn't just a dower gene.
Exactly. It was a direct, powerful regulator of adult longevity itself.
And they meticulously retested the fertility question with DAF2 mutants,
Sure.
just to be absolutely sure, right? The skepticism from the field must have still been immense.
Oh, absolutely. Scientific rigor demands you keep questioning. So they found that even when they ablated the reproductive precursor cells of DAF2 mutants, the animals still live long.
Okay, definitive again.
Yeah, this definitively confirmed that the extended longevity of DAF2 mutants was independent of any differences in fertility, thoroughly reinforcing the idea that it was a true autonomous longevity mechanism.
So DAF2 was important, but the puzzle wasn't complete. Then came DAF16, which was already known to interact with DAF2 in dower formation. How did they connect DAF16 to adult longevity?
Right. The DAF2 gene was already known to require another gene, DAF16, for dower formation. So the logical next step was to ask, is DAF16 also required for the extended lifespan of adult DAF2 mutants?
Makes sense.
And apparently, with no students immediately available for this crucial experiment, Kenyon, with her characteristic dedication, performed the experiment herself.
Wow. That's true dedication. And what did she find? What was the relationship between DAF2 and DAF16 in regulating lifespan?
She found that DAF16 was completely required for DAF2 mutants to live long.
Completely.
Completely. This was a monumental finding because it illuminated the core regulatory interaction. It meant that these two genes had analogous effects on dower formation and adult aging. Think of it like this. Wild-type DAF2 acts like a brake,
Okay.
and DAF16 is like an accelerator for longevity.
Got it.
Normally, DAF2 keeps DAF16 in check, preventing dower formation and limiting adult lifespan. But when DAF2's activity is reduced, that brake loosens and DAF16 is unleashed, promoting both dower formation and, critically, adult longevity. They even affectionately nicknamed wild-type DAF16 Sweet 16.
Sweet 16, I like it. Because it promotes youthfulness and extends life.
Exactly.
This sounds like the first really clear indication that genes encoding nutrient sensors were directly regulating aging. How did they make that connection, especially with food limitation?
Well, the connection emerged pretty naturally from the known role of DAF2 in dower formation. Low food conditions were already known to reduce DAF2 activity, which in turn triggered dower formation.
Right.
The hypothesis was that DAF2 mutations might just be mimicking an internal signal generated by food limitation. Their paper from 1993 even explicitly stated, it would be interesting to learn whether lifespan extension caused by food limitation requires DAF16.
And did it?
It indeed turned out to be the case, at least for some dietary restriction methods, like intermittent feeding. DAF16 was shown to be absolutely required for the full life-extending effect of those interventions.
Okay, so they had DAF2 inhibiting DAF16 working together, but what about Tom Johnson's original age-1 gene? Was it part of the same pathway, or was it a completely separate mechanism?
That was the next crucial piece of the puzzle for getting a unified understanding. Jenny Dorman, who was a technician in Kenyon's lab, set out to build the double DAF16 H1 mutant.
Okay.
And the striking and exciting result, as they put it, was that this double mutant was not long-lived.
Ah, so age-1 needed DAF16 too.
Exactly.
Yeah.
This was incredibly important because it meant that age-1 was indeed part of the same pathway as DAF2 and DAF16. This finding wasn't just scientifically profound, showing this unified genetic pathway for longevity, but it was also practical. It was crucial for the subsequent cloning and molecular identification of age-1.
And this convergence led to identifying age-1 as DAF23. How did that happen?
Yes. The observation that DAF16 suppressed age-1's longevity strongly suggested that age-1 might just be a weak allele, a slightly broken version of a known dower gene.
Okay.
The gene DAF23 was a prime candidate because it behaved genetically similarly to DAF2 and was being actively studied by Gary Rovkun's lab. Then Jim Thomas's lab, motivated by Kenyon's team's unpublished results and just the growing excitement, found that H1, H by 5546 mutants actually became dowers at a high temperature, 27 degrees C.
Ah, a visible phenotype.
Exactly. This visible phenotype allowed them to easily map H1 and confirm it was the same gene as DAF23. Rifkun's group independently confirmed this too, solidifying all his genetic connections.
Okay, so now they had DAF2, DAF23H1, and DAF16 all linked together in a pathway. The big question then became, what kinds of proteins did these genes actually make? This is where the molecular identification truly reveals the groundbreaking nature of the pathway, right?
Precisely. The molecular identification of these genes was the ultimate game changer. It moved beyond just genetic interactions to the underlying biochemistry. Gary Rifkun's lab first cloned DAF23H1, revealing it encoded a phosphatidylinositol 3 kinase, or PI3 kinase.
PI3 kinase. Okay, what does that do?
So for listeners, a PI3 kinase is basically a crucial enzyme that acts like an internal relay station. It transmits signals related to cell growth, metabolism, and survival inside the cell. Now, while PI3 kinases were known to signal downstream of various receptor tyrosine kinases, at that specific time, no one directly linked this particular one to insulin IGF-1 signaling specifically.
Why not?
Well, PI3 kinases are involved in many pathways, so the specific connection wasn't immediately obvious just from identifying it as a PI3 kinase.
Okay, so it wasn't a sudden aha moment for the entire pathway then, just one crucial piece of the biochemical puzzle. The truly stunning finding for the pathway, and arguably the biggest breakthrough, came with identifying DAF2.
Yes, that was the truly stunning finding that unified everything. In 1997, Rifkun's lab reported that DAF2 encoded the C. elegans homologue of the human insulin and IGF-1 receptors.
Whoa, the actual insulin receptor.
Essentially, yes, the worm version of it. This was monumental. It meant that hormones, evolutionarily conserved hormones like insulin and insulin-like growth factor 1, directly controlled aging. This suddenly made immense sense from the perspective of dower formation, because insulin IGF-1 signaling is such a fundamental pathway regulating growth, metabolism, and nutrient response across species.
Okay, here's where it takes that really wild and kind of funny turn, the initial skepticism from the medical community. I remember hearing about Morris White, a leading insulin signaling expert, famously telling Kenyon at a diabetes meeting something like that such mutations would never extend the lifespan of a mammal, they would just cause diabetes.
Hey, yeah, that quote perfectly captures the immense paradigm shift that was happening. It was incredibly jarring for many to even conceive that inhibiting such essential signaling pathways, known for their critical role in metabolic diseases like diabetes, could actually slow aging. It just challenged deeply held assumptions.
But he came around, didn't he?
He did. To his credit, Morris White later became a friend and, significantly, reported himself in 2007 that mouse mutations disrupting the insulin and IGF-1 pathway gene IRS2 did extend lifespan.
Wow.
Albeit, yes, with increased blood glucose levels. But this landmark study showed the mammalian relevance and suggested that maybe reducing insulin IGF-1 signaling acts as a kind of danger signal, shifting physiology towards cell protection and maintenance, rather than just causing disease outright.
Fascinating. And the final piece of the puzzle, DAF16. What was its molecular identity, and why was that so critical for understanding how longevity was being regulated at the genetic level?
Right, DAF16. Both Gary Rifkun's and Kenyon's labs independently cloned DAF16 in 1997. DAF16 protein was identified as a forkhead family, or FOXO, transcription factor.
Okay, and a transcription factor is?
It's essentially a master control protein that switches specific genes on and off. So DAF16 was orchestrating the cell's response to stress, influencing metabolism, and promoting longevity by activating this whole cascade of protective genes.
Ah, the conductor of the orchestra.
Exactly. This was, as Kenyon described it, monumental for aging research. It provided a direct molecular mechanism. A transcription factor could promote lifespan extension. There was no longer really any doubt that aging was a regulated process, and now there was a clear path towards understanding the mechanisms of longevity at the molecular level by figuring out DAF16's downstream targets.
So we went from organisms just wear out to evolutionarily conserved hormones and master control genes regulate aging. The impact must have been just explosive, completely reshaping the field.
Absolutely explosive. The study of insulin IGF-1 signaling and FOXO proteins in aging just blew up after 1997. We now know that C. elegans lifespan can be extended an astonishing 6 to 10-fold by targeting this pathway.
Tenfold.
Tenfold. More importantly, this pathway's influence on lifespan has now been shown in flies and mice, demonstrating profound evolutionary conservation. There are even hints of its role in natural evolution, like Smaller dog breeds, for instance, which are often IGF-1 mutants, tend to live longer than larger dogs.
Huh.
And circulating IGF-1 levels are inversely correlated with lifespan across different mouse strains. It seems to be a truly universal pathway.
And the human connection is maybe the most exciting part, right? Suggesting these ancient pathways are still working in our own longevity.
Indeed. Impaired IGF-1 receptor activity has actually been linked to centenarianism in Ashkenazi Jews, and specific FOXO DNA variants have been associated with exceptional longevity in incredibly diverse human populations. Hawaiians of Japanese descent, New Englanders, Germans, Italians, Ashkenazi Jews again, and the Chinese.
Wow, that's broad.
It is. Now, the precise mechanisms of how these specific variants affect gene activity are still being figured out, but these connections just underscore the pathway's profound relevance to human longevity and health span.
And beyond just living longer, what's really compelling is the broad protection against age-related diseases that these pathways seem to give. It's not just lifespan, it's health span.
Yeah, what's truly fascinating here is that many of these long-lived mutants aren't just surviving longer. They're resistant to a whole spectrum of age-related diseases. Cancer, heart disease, protein aggregation diseases like Alzheimer's or Parkinson's. This suggests the incredible potential to maybe forestall multiple diseases simultaneously by targeting aging itself, rather than trying to address individual diseases one by one after they appear. This foundational work truly laid the groundwork for the entire field of longevity research as we know it today. It shifted our understanding from passive, unavoidable decay to a finely tuned, genetically regulated, and crucially, a malleable system. It gave us permission, really, to believe that aging could be intervened with.
That was an incredible journey through a truly foundational paper. Kenyon's work really showed us that aging isn't some fixed fate, but a process we can actually understand and potentially control. So with that historical context firmly in mind, the initial skepticism, the painstaking detective work, and that ultimate paradigm shift, let's turn our attention to some of this week's papers. Even if they represent more incremental steps, they're building on the shoulders of giants like Kenyon, right? And we can apply that same critical lens to see their true potential.
Absolutely. It's the daily grind of science, these incremental findings, that eventually builds towards the next paradigm shift. Each paper, even if it feels small, contributes a piece to the larger, ever-evolving puzzle. We learn from the past to better interpret the present.
Okay, let's dive into the first one. The aging factor EPS8 induces disease-related protein aggregation through RAC signaling hyperactivation. This work comes from Seda Koyuncu, Yaiza Dominguez-Canterla, and Rafael Alice at the University Hospital Cologne, published just last Thursday, September 4th, in PubMed. So what did they find, and why is it important in this broader context of age-related diseases?
Right, so this paper tackles a really significant problem. Aging is the #1 risk factor for a whole host of devastating neurodegenerative diseases, things like Huntington's, ALS, even some forms of Parkinson's. And a common, grim hallmark of these conditions is the pathological aggregation, the clumping up of misfolded proteins inside neurons.
Right, the protein clumps.
Exactly. Now, while these diseases involve different specific proteins, polyglutamine repeats in Huntington's, mutant FUS, and TDP43 in ALS, their common late onset strongly suggests they might share some convergent aging-related changes that actually trigger this aggregation process.
Okay, so if I'm understanding this correctly, EPS8 RIC signaling basically acts like an accelerator, or maybe like an aggregator hub for these terrible protein diseases as we age.
That's a good way to put it.
And if you target it, you could potentially slow down that acceleration across different diseases.
That's exactly the idea. The key discovery here is that age-associated hyperactivation, so too much activity, of this EPS8-RAC signaling pathway in C. elegans, actively promotes the pathological aggregation of these disease-related proteins. They specifically showed this for Huntington's polyglutamine repeats and for those ALS-associated mutant FUS and TDP43 variants.
Huh.
It's like discovering a faulty switch that, when it gets stuck in the on position with age, causes these diverse proteins to start clumping together. And they found that knocking down the gene for EPS8 or its downstream RAC orthologs actually prevented this protein aggregation and the subsequent neuronal problems in aging worms.
That's pretty compelling. That didn't just stop at worms, right? They moved into human models, too.
That's right, and that's a critical step, obviously. They showed that inhibiting EPS8 signaling also reduced aggregation and neurodegeneration in human cell models. That provides some crucial translational relevance beyond the worm.
Okay.
Furthermore, they dug into the mechanism, the how. They identified this ubiquitinating enzyme, USP4, as a key regulator of EPS8's ubiquitination and degradation.
Okay, wait. Ubiquitinating enzyme, USP4. What does that mean in simple terms?
Right, sorry. So ubiquitin is like a little tag cells put on proteins to mark them for destruction. Ubiquitinating enzymes, or DUBs, basically take that tag off, which stabilizes the protein and prevents it from being degraded.
Ah, okay, so USP4 keeps EPS8 around.
Exactly. They found that reducing the age-related upregulation of USP4, so preventing USP4 levels from rising too high with age, not only prevented EPSA from accumulating, but remarkably also extended longevity and reduced these disease-related changes in the worms.
Wow, longevity too.
Yeah. So USB4 acts like a brake on EPS8 accumulation, and if that brake fails with age, EPS8 builds up and promotes all that nasty protein aggregation.
Okay, that's a lot of intricate molecular detail, but the big picture takeaway seems to be a new, potentially convergent target for multiple neurodegenerative diseases and even a hint of longevity extension. Why is finding a convergent factor like this so intriguing?
Well, what's really intriguing here is exactly that EPS8 appears to act as a convergent aging factor. Instead of needing to develop, a different drug for Huntington's protein, another for ALS protein, finding a common pathway, a single upstream factor like EPS8, offers this really tantalizing possibility of a broad therapeutic approach.
One drug, multiple diseases.
Potentially, yeah. That could tackle multiple conditions simultaneously. That's kind of a holy grail in drug discovery for complex diseases. And the identification of USP4 as a regulator of EPS8 degradation also offers a specific, potentially druggable target. DBs like USP4 increasingly recognize as viable drug targets because they're enzymes, which makes them generally more amenable to small molecule inhibition, something pharmaceutical companies like.
Okay, so a convergent factor and a potentially druggable enzyme target. That does sound promising. But of course, we need to apply our rigorous skeptical lens here, especially given the challenging history of neurodegeneration research. What are the crucial caveats we need to keep in mind?
Yet several points definitely warrant some caution. First, while they did include some human cell model validation, which is good, this work is still primarily focused on C. elegans.
Right.
And translating findings from worm longevity or disease models to mammals, let alone humans, is notoriously difficult and expensive. It's a huge hurdle. Many incredibly promising targets from worms, flies, even mice just failed to show efficacy or safety in humans. The physiology is just way more complex.
Right. You definitely don't want to fix one problem by creating 10 others,
Yeah.
especially with a fundamental protein like EPS8 must be.
Exactly. That brings us right to the second point. EPS8 itself has crucial physiological functions. It's involved in cell motility, growth, general cellular signaling. It's not some obscure protein. It's a really fundamental part of cell biology. So broad systemic inhibition of EPS8 could potentially lead to severe, undesirable side effects, disrupting normal cellular processes all over the body. Figuring out how to specifically inhibit its aging-promoting activity without messing up its essential functions would be a major, major challenge.
And the history of trying to develop drugs for protein aggregation diseases is not exactly filled with resounding successes, is it?
Far from it. That's our third caveat. The field of protein aggregation has a long and, frankly, disheartening history of promising targets, especially in Alzheimer's and Parkinson's, that have just failed spectacularly in clinical trials despite looking great in preclinical models. It's a notoriously difficult area. Often it's due to the complexity of protein misfolding itself, maybe the irreversible nature of aggregation once symptoms actually appear, or just unforeseen off-target effects of the drugs. So yeah, a healthy dose of caution is always warranted given past experiences and the sheer challenge of these diseases.
And the longevity data.
Right. Finally, the lifespan extension data is reported in worms. While that's encouraging, it really needs robust validation in mammalian models to be considered truly impactful for human longevity. We've seen so many longevity genes in worms that just don't translate to significant lifespan extension in mice or humans.
Okay. So with all that in mind, what's next? What experiments would you really want to see to move this forward convincingly? What would constitute real progress here?
Okay, to really validate this, first and foremost, we absolutely need comprehensive toxicity studies of EPS8 inhibition in mammals. It's critical to understand the potential side effects on the brain, heart, liver, you name it, before even thinking about any human application.
Safety first.
Always. We also need a clear demonstration that this approach actually extends mammalian lifespan, not just prevents specific disease models in a dish or a worm, showing that it delays onset or slows progression of neurodegenerative diseases in higher organisms, mice ideally, maybe even non-human primates, for really robust validation. That would be a huge step.
Okay.
And of course, more robust validation of the convergent mechanism across multiple different neurodegenerative disease models in higher organisms is essential. Proof is not just a worm-specific thing or limited to only one type of aggregation. This would involve specific assays looking at, cognitive function, motor skills, and direct measures of protein aggregation in actual brain tissues.
Very clear. A promising lead, definitely, but yeah, a very long road ahead. Okay, let's move on to our second paper of the week. Platelet factor 4, PF4, regulates hematopoietic stem cell aging. This fascinating work comes from Sen Zhang, Charles E. Ayamoba, and Anna M. DiStallo from the University of Illinois at Chicago. It was published very recently, just this Monday, September 8th, in PubMed. So what did this team discover about PF4 and its role in aging?
Right, so this paper tackles another really critical aspect of aging, the decline of our hematopoietic stem cells, or HSCs.
And blood stem cells.
Exactly. These are the remarkable master cells sitting in our bone marrow, responsible for producing all our blood cells, red blood cells, white blood cells, all the various immune cells. And as we age, HSCs and their specialized little homes in the bone marrow, their niches, undergo these detrimental changes. This leads to immune decline.
Making us more susceptible to infection.
Right, and also an increased risk of blood cancers, like leukemia.
So basically, our blood-producing factory gets old and tired, leading to problems throughout the body. What did the researchers find was causing this decline?
Well, the key discovery here is that age-related alterations in the megakaryocytic niche, that's a specific microenvironment in the bone marrow that supports HSCs, made by megakaryocytes.
So platelet-making cells.
Exactly those.
Hmm.
Changes in that niche and critically a significant downregulation, so less of, a protein called platelet factor 4, or PF4, seem to be pivotal mechanisms driving HSC aging. PF4 is this small protein secreted by those megakaryocytes. So they essentially found that as we age, the bone marrow niche changes, and importantly, there's just less PF4 around.
And they showed this by looking at mice that lacked PF4. What exactly happens to their stem cells?
That's right, they created PF4 deficient mice, and these animals showed several phenotypes that looked a lot like accelerated HSC aging. Specifically, they had lymphopenia, a decrease in crucial immune cells called lymphocytes. They had an increased output of myeloid cells at the expense of lymphoid cells, which is a really common shift we see in aging immune systems. And they had increased DNA damage in their HSCs. These are all classic hallmarks that mimic naturally aged HSCs, strongly suggesting PF4 normally plays a protective role. But here's where it takes a truly exciting turn. The rejuvenation aspect.
This is the part that really caught my eye. Rejuvenation. That's a powerful word, often a controversial one. In longevity research, what did they mean by that?
It is a strong word, and they used it with good reason here, I think. Remarkably, they found that administering recombinant PF4, so basically just giving back the missing PF4 protein-restored old HSCs to youthful functional phenotypes.
Really? Youthful? How?
Well, this included several pretty striking improvements. The aged HSCs showed improved cell polarity, which is crucial for proper cell division and function. They had reduced DNA damage, a hallmark of youth. They exhibited enhanced in vivo reconstitution capacity.
Meaning they could actually repopulate the blood system better?
Exactly. These rejuvenated cells could more effectively repopulate the entire blood system of a recipient animal. And they also achieved a more balanced lineage output, correcting that myeloid bias seen in aged HSCs. This truly suggests a reversal, not just a slowing down of aged phenotypes. It really is potentially a game changer.
Wow, actual rejuvenation of fundamental stem cells. That's incredibly impactful. And did they dig into the mechanism? Did they identify the specific receptors involved?
Yes, which is vital if you want to develop targeted therapies. They identified LDLR and CXCR3 as the HSC receptors that actually transmit the PF4 signal.
Okay.
And to confirm this, they engineered double knockout mice lacking both of these receptors, and those mice showed exacerbated HSC aging, looking very similar to the PF4 deficient mice. This really reinforces the mechanistic understanding. It tells us exactly how PF4 is talking to the stem cells, and crucially, they also observed that human HSCs across various age groups also responded to this youthful PF4 signal.
But in cells, too.
Yes. This direct evidence in human cells is a very strong positive indicator for its translational potential. It shows it's not just some weird mouse-specific phenomenon.
Okay, that's why it's so exciting then. Actual reversal of aging in critical stem cells, detailed mechanistic understanding, and direct human relevance. It really sounds like a potential game changer for tackling immune decline and maybe even cancer risk in aging.
It genuinely does offer some tantalizing possibilities. I mean, the ability to reverse age HSC phenotypes back towards youthful states could have profound implications for immune function and overall health in older individuals. especially considering that HSC aging is such a major contributor to immune senescence that increased susceptibility to infections and the higher risk of blood cancers. Imagine boosting the immune system in the elderly back to something resembling that of a younger person. The impact could be enormous.
All right, now for the other side of the coin, our rigorous skeptical lens. This sounds almost too good to be true sometimes, so what are the crucial caveats we need to consider here?
Right, okay. First, while the functional assays showing rejuvenation are impressive, much of this work, particularly the most dramatic effects, is likely derived from ex vivo assays, meaning cells tested outside a living organism, in a dish, or maybe short-term in vivo studies.
Okay.
We really need to see if chronic systemic PF4 treatment actually extends lifespan in vivo in mice. That's still the ultimate gold standard for any real longevity intervention. The effects you see in vitro or in short-term challenges don't always fully translate to long-term whole organism benefits.
And PF4 is known for its role in blood clotting, right? That immediately raises flags for giving it systemically.
It absolutely does. That's our second and perhaps most critical caveat. PF4 has known complex physiological roles in hemostasis blood clotting, as well as in inflammation and immune function. Systemic administration of PF4 could lead to serious, potentially dangerous side effects. You could alter blood clotting pathways, leading to thrombosis, or maybe even bleeding issues. We have to be very cautious about off-target effects and make sure any therapeutic window is wide enough for safe application.
So the balance between benefit and risk is still a huge unknown, and the magnitude of these benefits needs to be rock solid validated.
Precisely. Third, the magnitude of rejuvenation, while it looks impressive in these functional assays, needs to be validated in longer-term whole animal studies. We need to understand if these youthful phenotypes actually persist over time and truly translate into sustained health benefits, like, actually reduced infection rates or lower cancer incidence in aged animals.
Right.
And finally, we just have to learn from history. We've seen similar initial excitement around other young blood factors like GDF11, remember that?
Oh yeah, GDF11, big splash initially.
Huge splash, showed great promise in early studies of muscle and brain rejuvenation, but later had really mixed and often unreplicated results. The scientific community engaged in robust debate, highlighting the challenges of these complex systemic factors. We absolutely need robust independent replication here with PF4 to build confidence and move forward safely.
Absolutely. Lessons from history are incredibly important to maintain our scientific integrity. So for the team at the University of Illinois, Chicago, what are the next critical experiments that would truly solidify these findings and maybe pave the way for potential translation?
Okay, to move this forward convincingly, we absolutely need studies demonstrating chronic systemic PF4 treatment that results in actual lifespan extension in mice. That would be the definitive proof of concept for a longevity intervention.
The big one.
The big one. Alongside that, comprehensive safety profiling for systemic PF4 administration is paramount. We need to fully understand its effects on the entire physiological system. Coagulation panels, cytokine profiles, long-term organ function, everything. And finally, validation that this HSC rejuvenation truly translates to significantly improved immune function, maybe measured by enhanced vaccine response or reduced susceptibility to specific infections, and a genuinely reduced cancer risk in aged animals is essential. That would be the real proof of concept for its therapeutic potential in human aging.
What an extraordinary discussion this week, stretching all the way from foundational discoveries that literally redefined aging to these cutting-edge, maybe incremental insights that hint at the future.
Indeed. We revisited a true cornerstone of longevity research with Cynthia Kenyon's groundbreaking work on the insulin IGF-1 pathway in C. elegans. That research just fundamentally shifted our understanding of aging from, passive decay to a beautifully regulated, genetically malleable process. That historical context really highlights the incredible journey from that initial scientific skepticism to a global research phenomenon.
Wow.
It just illustrates the courage and dedication required for true paradigm shifts.
And then we fast forwarded to this week's papers, exploring two really fascinating areas. The EPS8-RSC signaling pathway emerged as a potential convergent factor in neurodegenerative protein aggregation, offering new ways to understand and maybe eventually treat diseases like Huntington's and ALS.
Right.
And then there was the remarkable potential of platelet factor 4 to actually rejuvenate aged hematopoietic stem cells, which truly holds the promise of reversing certain aspects of aging, at least in the immune system.
Both offer these tantalizing possibilities, right? A glimpse into what might be possible in the future. But as always, the path from initial findings in model systems and ex vivo results to clinically viable, safe therapies for humans is incredibly long and fraught with challenges. It's just crucial to balance that hope and excitement with a healthy dose of scientific skepticism regarding translation, especially when dealing with such complex biological systems.
Yeah, I think the continuous journey is the key takeaway from today. It's this constant interplay between foundational discoveries that redefine the field and the iterative, often incremental progress that follows. Kenyon's work showed us aging is malleable. It's a process we can understand and potentially control. Now, the grand challenge lies in translating those profound insights into practical, safe interventions that genuinely impact human health and lifespan, moving from possibility to reality.
Precisely. And as we continue to uncover these intricate molecular pathways and chip away at the mysteries of aging, a critical question starts to emerge for all of us, doesn't it? If we can indeed extend youthful function across multiple organ systems and potentially push the boundaries of human health span, how will our understanding of what it even means to age, both biologically and culturally, fundamentally transform? Will we end up redefining what it means to be old entirely?
What stands out to you from today's discussion about this interplay between groundbreaking science and the persistent need for skepticism? We'd love to hear your thoughts. Thank you for joining us on the Deep Dive. We'll be back next week with more deep dives into the latest research.